Hi, Liz.
Hi, Michelle. Hello, Raptors.
Hi, Raptors. All right, we're hitting podcast number 3, Alzheimer's disease today. Hits extremely close to home for you and I, Liz, since that's the primary reason I think we're taking rapamycin. And of course, the prevalence in our family history of Alzheimer's. And I think it's one of the primary reasons a lot of people seek out rapamycin therapy as well, the APOE4 status. So again, Dr. Green breaks down what Alzheimer's is. And my takeaway is that there's nothing that we can do these days to treat, nothing's treating it effectively. The best course of action is prevention, and rapamycin is the best method to do that.
Yep, as far as we know. Yeah, so during the podcast, Dr. Green talks about amyloid beta and tau as being a byproduct of Alzheimer's disease and not the cause. That should be becoming pretty clear with all the 30 years of studies and drug development that have targeted amyloid beta and tau and have been unsuccessful. In 2011, a person named George Bartzokis wrote a paper which is consistent with Dr. Green's beliefs on what causes Alzheimer's disease. And basically, the cause is the age-related inability to maintain the cerebral microvascular system and myelin. Humans are the only animals that get Alzheimer's disease. And, we have a big super computer brain that requires lots of glucose and oxygen. And basically, it breaks down over time. And people with the APOE4 gene are particularly susceptible to that. And rapamycin is very good at preventing Alzheimer's disease. There are even some studies in mice. Now, I know I just said mice don't get Alzheimer's disease, but they do program them to get something similar to Alzheimer's disease. And they've been able to show that these mice given rapamycin have been able to either prevent and even there's been some improvements that have already have it. So, but the best course is definitely prevention. It's currently not considered a treatable disease, and so Dr. Green explains this and goes over George Bartzokis' study. It's pretty detailed, and I definitely didn't follow everything, but the gist is that there's a different cause, and that rapamycin is effective in preventing that cause, especially the increased exposure that APOE4 genetic carriers have. So
cool.
Without further ado, we'll go on to Dr. Green.
Here's Dr. Green.
So the most important thing for people to know about Alzheimer's disease is that it's a preventable disease. It's not a treatable disease. In this way, it makes it very similar to most other steroid diseases. When you have an end-stage heart disease or liver disease or lung disease, those diseases are not treatable. Those people require an organ transplant. The problem is you can't have a brain organ transplant. So rapamycin, like Alzheimer's disease, has to be prevented. Preventing Alzheimer's disease requires knowing a whole lot about what's happening and especially who's at risk. I'm going to start the discussion with comments about the amyloid hypothesis. To borrow a line from Mark Antony, I come to bury the amyloid hypothesis, not to praise it. Anyway, the amyloid hypothesis has dominated thinking about Alzheimer's disease for the last 30 years. I consider it one giant rabbit hole. But the problem with it is all the money for the last 30 years has gone into the amyloid hypothesis. Brilliant scientists who realized that amyloid was not the cause of Alzheimer's disease weren't able to get on the amyloid bandwagon, weren't able to get funding, weren't able to do research, were pretty much forced out of the field. That was the problem, that it dominated all this funding. Basically, it's been known for over 100 years that end stage of Alzheimer's disease was characterized by amyloid and tau. It was somehow in the last 30 years, they came up with the idea that this was the cause of the disease, as opposed to this was the end stage of the disease. The reality is amyloid and tau are the downstream byproduct of the upstream disease process. So why I sort of like mention this is, in my opinion, the greatest researcher about Alzheimer's disease in the last 20 years was George Bartzokis. He wrote a spectacular paper in 2011 explaining very much how Alzheimer's disease worked. The paper got very, very, very little attention because it did fit in with the amyloid hypothesis. Anyway, so George Bartzokis tragically died around 2014. He was like a researcher at UCLA. He was like a giant in the field of understanding Alzheimer's disease. So his last paper was, we'll go over it. The name of the paper was Alzheimer's Disease as a Homeostatic Response to Age-Related Myelin Breakdown. Complicated topic, but we'll sort of explain what he's talking about. Let's see like the first diagram. Yes. Yeah, so this is the first diagram. And in this diagram, what he's trying to show that myelization reaches sort of like a peak. This is all these little dots of myelization. This one is an MRI. This was like an autopsy. He's showing that myelization like gradually builds up, reaches a peak at like age 45, and then gradually declines. So that's like myelization in the frontal lobe in the white matter. In other words, this is the myelization that's going on within the actual sort of like between the neurons. So basically, Bartzokis is coming up with what could be called the myelin theory of Alzheimer's disease. The myelin theory of Alzheimer's disease is basically the idea that what's special about the human brain is myelization. Myelization in the cortical areas of the frontal lobes, like not myelization going from the brain to sort of like peripheral tissues, or the brain to the eye, or the brain to the legs. Myelization going from one neuron to the other neuron in the gray matter. Why myelization is so important is myelization increases the speed like 20-fold. The idea of a supercomputer is to make a supercomputer, you need speed, and you need millions and millions of connections. The human brain was not a scaled up monkey brain with more neurons. It was a brain that had like proportionally the same amount of neurons, but the areas were like intensely myelinated in the cortical areas, and they had very, very, very more connections. So this myelization is what made the human brain unique, what made the human brain a supercomputer, the speed and all the connections. And it's what made the human brain susceptible to Alzheimer's disease. It turns out that humans are the only animals that get Alzheimer's disease And that's because they're the only animal that has all this myelization into sort of like cortical areas. So that's the sort of like, so that's the idea. That's the critical aspect. So the difference is understanding Alzheimer's disease starts with the idea of understanding why is the human brain different than all other brains? What makes it a supercomputer? The understanding that it's the myelization, and then understanding how the myelization leads to Alzheimer's disease. The problem with the myelization, now the human brain requires a huge, huge amount of energy. It gets 25% of the entire energy that the body produces. That's like measured by 25% of the oxygen utilized and the glucose. This is enormously more than any other animal. So the humans made a strange kind of bet. They bet on giving their brain a huge, huge blood supply, and they somehow thought that was going to pay off. It turned out to be a very good bet. Okay. But the problem with the supercomputer is it's one thing to build it, it's another thing to maintain it. Okay, let's do our diagram 2. This diagram takes a lot of explaining. What it is a neuron that's been cut in half to show the inside of it. Here's the sort of like head of the neuron's sort of head. Here's the synapses, and this all part would be the axon. Here's the oligodendrocyte, which is maintaining the myelization, and it's been cut open. And when it's been cut open, what's revealing is something called fast axon transport. The top figure is like normal. Fast axon transport is how the neuron maintains itself. So the neuron is proportionally a huge, very, very, very long. It has the neuron at one end, the synapses at the other, and in the middle is a huge, huge distance. And you have to have things that are made up here at the neuron. They have to go to support the synapses, and the synapses have to communicate with the neuron. And how this is done with something called fast axon transport. And what fast axon transport is, in the center of the neuron, at the center of the axon, there's these little railroad tracks. So these are sort of like, here's like a railroad track. Here's a railroad track. The railroad tracks go, they have little cars on them. These cars go that direction. These cars go this direction. And they carry things back and forth from the neuron to the synapses. And what it looks like they're carrying here is mostly mitochondria. Here's like healthy mitochondria being carried to the synapses. Here is sort of like damaged mitochondria being carried back to the nucleus to repair. Here's some other particles. But you could notice that they're running on these two lines. One is a gray line, and one has all sorts of green little hatches going across. The green little hatches are tau. It turns out that tau is microtubules that stabilize the tracks that these little cars are running on. That's the function of the sort of like tau. Tau is basically parts of like railroad tracks. People like with the here they have little green spots going across horizontal to the main line. These are sort of like, this is tau, and this is stabilizing the tracks, which the little cars are running up and down on to carry things. So this is a healthy, normal functioning like axon. This other one is an axon that's undergone repair. And this is the repair process. So the repair process is first it stops the transport. So the tau becomes, as phosphate groups, becomes hyperphosphorylated. That stops the tracks. Breaks up the tracks. Little cars fall off the tracks. They can't transport things anymore. And in this area is now, it's been being repaired. So it's under repair. At this time, the sort of like synapses fall apart. And now you have this repair process going on. And in this repair process, the things that are taking part in the repair process includes amyloid and what's called BACE and amyloid precursor proteins. These are all these sort of like little dots and so forth that they're given. These are all little bits of amyloid, and they're taking part in the repair process. That's what the amyloid is doing. It's basically, the amyloid itself is sticking these little particles onto little cars. So this is the repair process going on, and this would be a swelling, and this would be a neuron that's now trying to repair itself.
Dr. Green, I have a question. What is it trying to repair? What's happened?
Trying to repair the myelin. So the myelin is broken down, and it's trying to repair the myelin, and the oligodendrocyte, that's the cell that covers these things, and they're the cell that's what makes the myelin. And the whole process, the idea is to repair the breakdown of the myelin, which covers the neurons.
And why is the myelin damaged?
It could be damaged for a lot of reasons. It's basically damaged because it has to be repaired. The myelin has a tendency to break down. It has to be repaired. It's sort of like, that's the problem. That's what's going on. A repair.
Is that because of senescence, Dr. Green? Is this an aging related dysfunction?
This sort of like things of simply repairing maintaining the myelin as be considered a normal process.
So I guess normal function for the body's myelin to break down to some degree and repair itself.
Okay, maybe I'm jumping ahead though, because I'm looking at the myelin sheath in the second. Is that indicating?
This is an example of trying to sort of like repair the myelin breaking down. If we go to the next diagram, it becomes clear what they're trying to show.
Okay, but in any event, it seems like this is a normal process that would occur without having Alzheimer's disease. A normal process. This happens at all ages. Like when you're a child, you can have myelin breakdown too, or it's like a normal maintenance function like we have in other parts of our bodies.
Children aren't very much myelinated. They're not having myelin breakdown. Myelinization is coming to place in like when you get older, and the repair of the process is taking place when you're older. So this is like maintaining the myelin. But the critical thing to see is that there's inside the cell. Okay, here's the next sort of like thing. Here's sort of like there's a schematic diagram of trying to repair what they just called myelin breakdown. So now they're saying what the thing could contribute to myelin breakdown is age, free radicals, toxins, different things could cause myelin breakdown. As soon as you have myelin breakdown, you go into the repair process. The repair process then starts with right here, hyperphosphorylation of tau. As soon as that happens, it stops what was called fast axon transport. And then you get this axon swelling. And now you have this whole sort of like process of trying to repair the myelin. And here they're saying the things that could contribute to repair is being APOE4 or 3 and having the right amount of cholesterol and sulfatides and having low free radicals and having low inflammation, and those type of things sort of like improve your ability to repair the myelin.
Okay.
So the process either it might be successful or it might be unsuccessful. Usually the problem is probably, especially in younger people, is successful. The problem is what happens when it fails. So here they have failed repair. The thing that's going to contribute to failed repair is APOE4 and poor amount of cholesterol and inflammation and iron and free radicals that can contribute to failed repair. But the critical thing is what happens when the repair fails. When the repair fails, the swelling, which is the middle of the whole thing, bursts. When the swelling bursts, it releases amyloid peptides, which are part of the repair process, and it releases p-tau, hyperphosphorylated tau, which is the railroad tracks ties that were broken up to stop the transport, and a few other things that are released. And then it says the amyloid that's released then forms amyloid plaques. The tau causes neurofibrillary tangles. These contribute to the lesions you see in Alzheimer's disease, and it can contribute to the neuron death and the disability. And whenever this sort of like stops, you lose the sort of like memories. The point is, in this repair process, amyloid and tau is part of this repair process. It's nothing way. So that's what makes it not the cause of the disease. That's what makes it the secondary byproduct of the failed repair. When the repair fails, amyloid, the amyloid, iron, and tau, all that stuff that sort of like makes up the lesions you see in Alzheimer's disease are released. So this is basically the basic idea that Alzheimer's disease results in a failure to maintain the myelin, which then results in the fast axon transport repair fails. You get amyloid and tau in the lesions. And if you were able to repair it, you'll be fine. There's sort of like the axon will be fine. Okay. So that's sort of like, so that was the basic idea of sort of like Bartzokis, that the whole thing was related to myelin and maintenance of the myelin, and the cells that had the job of myelinization, maintaining the myelin, were the cells called oligodendrocytes. They're the cells that make the myelin. They could be called the hardest working cells in the body because of the large amount of myelin that they make. And these cells are susceptible to old age, and they're susceptible to inadequate blood supply and toxins. So, all right, so now the next part we're going to talk about is not this diagram, but the idea that Alzheimer's disease can be seen as two different types of diseases. It could be seen as a disease in young people, in middle-aged people who are APOE4 positive. And what's going on with these people is because they're APOE4 positive, they're having a much, much higher breakdown of the myelization, and in particular, a breakdown of the cerebral microvascular system, the blood-brain barrier, and the breakdown of the blood-brain barrier and the cerebral microvascular system is now sort of resulting in a failure to retain the myelization. That's one group of people. Then another group of people is old people who are not E4 positive. They don't have any special risk factors for Alzheimer's disease, aside from old age. And in old age, you just have a decreased function of the oligodendrocytes maintaining myelization. So basically, so age is playing a major role. Starting at about age 65, Alzheimer's disease is doubling. The risk is doubling every 5 years till it gets to about 90. And then the incidence is like 10% a year. And at 95, it's like 20% a year. And at age 100, it's like 40% a year. But the sort of like number of people with Alzheimer's disease in the older population is staying pretty constant because the people who don't have Alzheimer's disease are surviving nicely. People with dementia are dying rather quickly.
I have a question.
Yeah.
So aging is a significant risk factor for Alzheimer's.
What?
Aging is a significant risk factor for Alzheimer's, correct? Just age alone. So is that a greater risk factor than having an APOE4 allele?
Oh yeah, this sort of like the biggest risk factor is age. And as you see, age is sort of like doubling every 5 years, your risk is doubling. And by sort of like age 100, it's 40% a year. So basically, age is sort of like showing that Alzheimer's disease is basically represents the normal aging of the human brain.
So carrying an APOE4 allele just increases the risk further beyond age.
Right.
Correct?
The three risk factors for Alzheimer's disease. The first one is just being human. The second huge factor is age, getting older. And the third factor is APOE4. But I look at it as that two different conditions, and that in E4 people, it's presenting more like a disease in middle age. Things are going wrong because you're APOE4, not APOE2 or 3. However, in older people who are APO2 and 3, this is just the way that the brain is aging because it's not able to repair itself due to the high energy demands of maintaining all this myelination. So that's sort of like, so there is simply these cells, the oligodendrocytes, are breaking down because of age, as opposed to breaking down because they're being damaged. So, but in middle-aged people, the thing that's triggering Alzheimer's disease is a vascular disease. So then the disease becomes primarily a vascular disease, not a disease of old age. And it's the vascular disease that causes the breakdown of the cerebral microvascular system and the blood-brain barrier that's causing damage to the oligodendrocytes, which now means the oligodendrocytes aren't able to sort of like repair the myelin, and the whole sort of process is going on. But the whole process is going on because of a breakdown of the vascular system. The vascular system of the human brain is an absolutely extraordinary thing. It's like hundreds of miles of like capillaries, extremely close, producing this like incredible blood supply for the cells to be able to sort of like maintain this very, very high energy process that's going on. I mean, you can see the brain is 2 or 3% of the body mass, and it's using 25% of the energy. So it requires this huge amount of energy to do that, and that means a very good blood supply. And it also means not having a breakdown of the blood-brain barrier, because when you have a blood-brain barrier, toxins from the blood enter the brain, cause inflammation, very harmful. So this is basically explained. So I selected 4 papers, and the 4 papers are extraordinarily good papers for explaining what's going on from the point of view of the microvascular system causing Alzheimer's disease. So I'm going to go over those 4 papers. So I'll just read off the title. One paper was called but most of all these papers, they're for Veronica Galvin. So she was like the top researcher. And this paper is called mTOR Drives Cerebral Vascular Synaptic Cognitive Dysfunction in Normative Aging. So this paper is just normal aging. And what she looked at was normal rats. And she looked at normal rats. It's sort of like what age is sort of like a middle-aged rat, and then like 15 months old, an old rat. And they gave the rats rapamycin, and they saw what happened to their cerebral microvascular system. And it turned out that what happened is the rapamycin was able to maintain the function of the cerebral microvascular system in their old rats that were taking rapamycin. And what they described the findings, they said, is that the rapamycin prevented what they called hippocampal dependent learning and memory defects. What they were saying is that the memory was related to the hippocampus. The hippocampus is an area most vulnerable to decline. If the hippocampus is involved in short-term memory, the old rats were able to remember things when they did a water maze, and they said that was due to the mTOR, that resulting in mTOR, that rapamycin was reducing mTOR, and that's why they were able to remember what they were doing. It was able to retain memory. And then the next thing they said about the rats is that the vascular system was reactive. In other words, when they showed the rats some kind of like stimuli, it would change appropriately. So like basically it's the idea that when you think about something and you concentrate on it, that area gets a lot more blood supply. That's sort of like, that's the sort of kind of like, in other words, the kind of like functions, right? In other words, that the areas are getting a small blood supply, but as soon as you concentrate on something, or you look at it, or so forth, the sort of like reaction is to increase the blood supply to that area. And that was what was happening. So like that sort of reaction was being maintained. The other thing they showed it was maintaining what they call presynaptic density. In other words, this sort of like the density of the synapses. That was not declining with age. That decline is a function of losing the axons, the loss of the synapses. That was being maintained. And the other part was just related to the cerebral blood flow. The reaction of the cerebral blood flow was a normal reaction. So basically, the old rats treated with rapamycin, the cerebral microvascular system was reacting normally to different stimuli. The density was being maintained, the synapses being maintained, and the short-term memory related to hippocampus was being maintained. So this is in normal rats. And basically what they were saying is the first changes in Alzheimer's disease is a deterioration of the cerebral microvascular system. This going on in normal rats is sort of like an indication of, they were saying the rat's vascular system was the same kind of reactivity of the human one. So myelization was different, but the actual vascular system was the same kind of like reactivity. Another sort of like extremely important paper was also by Veronica Galvin, 2017 paper. And what she was saying was inhibition of mTOR protects the blood-brain barrier And they did this in models of Alzheimer's disease and models of vascular impairment. So they had two animals which represented that. They were giving them rapamycin. They were testing the integrity of the blood-brain barrier. The blood-brain barrier is being maintained. So they were saying that the blood-brain barrier was of extreme importance, but no, because it's keeping out toxins that are in the blood. But nobody knew how to maintain the blood-brain barrier. And this study was of extraordinary importance because they were showing that with rapamycin was maintaining the blood-brain barrier. And
Dr. Green, you said two animals. Do you mean two types of animals, like those mice with a rapamycin gene and those without? What do you mean by two animals?
This study was in mice, but they had a mice model of Alzheimer's disease and a mice model of vascular dementia. So that's what I mean, the two different animals. They had they were animals which had certain genetic defects, and that would make the genetic defect would result in vascular dementia or Alzheimer's disease. Those are so they were testing the blood-brain barrier in these two diseases, Alzheimer's disease,
vascular dementia. If mice don't get Alzheimer's disease, how do they use the APOE4 in mice?
The mice don't. The Alzheimer's disease is a sort of like progressive process. The mice get cognitive decline. They get decline of memory. They get decline of other things. They simply don't. They simply go to a certain level, and then they don't go further. In other words, in humans, it's this slowly deterioration of the myelin, which triggers an entire cascade of harmful effects. The animals that don't have all this myelin don't have this effect. So the animals can develop age-related decline in memory, age-related decline in function, but they don't get the severe dementia characteristic of humans.
Even with the APOE4 type of whatever they do to make rats be Alzheimer's rats, they don't get the same type of dementia?
We're going to be getting the full picture of Alzheimer's disease, because to get the full picture of Alzheimer's disease, you need to have myelinization. If they don't have myelinization, they can't get that. They can have deterioration. They can have an early stage of disease. They can bring the disease so far, they can see that they're having impairments, but they're never going to get to the stage of Alzheimer's disease with amyloid and tau and that kind of stuff.
Okay.
The other thing they sort of like found in this paper was the critical thing was that the reason that it was maintaining the blood-brain barrier, it was having an effect on like what they were calling tight junction proteins. So the rapamycin was maintaining tight junction proteins, and that was preventing the part of the deterioration of the blood-brain barrier. Okay. Now I'm going to get to.
So I have a question.
Yeah.
So if rats and mice don't get Alzheimer's and don't carry an APOE4 gene, how are we transferring this to the effect on humans with that gene and with Alzheimer's? Is it because it's an age-related? Is it because it's TOR?
What they can see in animals is sort of like in like wild type animals, like a normal, like rats, normal age. What they can see is a cognitive decline. The rats were not having Alzheimer's disease, but when they tested the rats swimming around, they were having problems remembering where things were. Their memory was impaired. And when they looked at the vascular system, it wasn't reacting well. It wasn't sort of like lighting up when they were looking at things. So they weren't so they could see there was an impairment of what was going on, but they never go into the stage of Alzheimer's disease, because in order to go to Alzheimer's disease, you have to have the breakdown of myelin. They don't have myelinated brains. They can't get Alzheimer's disease.
Okay, and that is why that's why no other animals besides humans have myelin. Is that correct?
Nobody else has the extent of myelization that would lead to Alzheimer's disease. They have some myelization, and they have myelization in other areas, but we're talking about myelization in the special areas, the frontal areas involved in thinking. They don't having myelization in those areas, and they're not having sufficient to cause the whole system to collapse.
Okay.
Now, another sort of like extraordinary important study was about something called cyclophilin A. This was a paper of extraordinary importance to E4 people. And what they were saying was that cyclophilin A is a pro-inflammatory substance. It can trigger the breakdown of the blood-brain barrier because its inflammatory response activates something called nuclear factor KB, and that activates something called MMP9, which is involved in all sorts of things. So MMP9 dissolves proteins, and that's dissolving to sort of like blood-brain barrier and causing the blood-brain barrier to sort of like become destroyed. So this is the idea of what it was doing. This was cyclophilin A, and cyclophilin A had to be in one compartment and not another compartment. It had to be in the brain compartment, not the vascular compartment. How it was going on was real complicated, but the point was it was cyclophilin A, the activation of cyclophilin A, which is now triggering the deterioration of the blood-brain barrier through activation of the end stage of like MMP9. It's like, which is like a very, very common proteinase that breaks down all sorts of things. And you have MMP9 in coronary disease, and it's breaking down. So like, you know, when the plaque is collapsing, it's like MMP9 is collapsing it. So MMP9 is a very common thing for dissolving proteins. And this is what's getting activated when the cyclophilin A was in the wrong place. And then what they said was there were three isoforms of APOE, E2, E3, E4. E2 and E3 were very good. E4 resulted in activation of cyclophilin A. And that was the problem with sort of like being E4. It was the activation of cyclophilin A, which then resulted in deterioration of your blood-brain barrier, which then resulted in toxins entering your brain, which then resulted in the oligodendrocytes beginning to sort of like fail, and the maintenance of myelin, and the whole cascade now going on. So it's like cyclophilin A. Now there was like one more fourth-page paper that was put the whole thing together. This is the paper that put everything together, and what they did was they made a transgenic APOE4 mice. So now they had a mice to ask you a question, well, what about E4? Now they finally had an E4 mice. In other words, they took the sort of like mouse, or like E4 mouse APO out. They put in a human APOE4, and now I can see what happened when you had a mouse with APOE4. And what happened was you had the expected things about the deterioration of like memory and vascular system and those sort of things. But this is the critical thing. it came down to the cyclopro, the CYPA that we talked about. So,
Oh, sorry.
so
Dr. Green, do you want this up here now?
no.
No, not yet?
No.
Okay. Well, let me stop sharing then. You tell me when.
So in this study, in the APOE4 mice, the cyclophilin A then became activated and it broke down the blood-brain barrier. However, rapamycin was able to stop this because it was able to stop the activation of the CYPA. So now you had exactly what was going on in the E4 people, from looking at the E4 mice, and the E4 people, that there was the activation of CYPA, resulting in breakdown of the sort of like microvascular system, and especially the blood-brain barrier. This is being blocked by rapamycin, and this is protecting the cerebral microvascular system. That's the sort of like thing that's going on. And the rest of the stuff that's going on in these sort of like the APOE mice is that
APOE4 mice.
APOE4 mice, they said rapamycin restored the vascular function of the APOE4 mice. And so they were saying there was a whole lot of up to like 38% loss of different amount of vasculature, like vascular density in different areas. Rapamycin was like preventing this loss of vascular density. And then they were saying it was restoring what they were calling the metabolic function. And then they were saying it was preventing the E4 mice from developing learning deficits. So they weren't developing learning deficits would be related to like the hippocampus, their blood function of the cerebral microvascular system, which is sort of like maintained in good order. But the critical thing about all this was that it was a CYPA. This was the sort of like remarkable finding that rapamycin was able to stop the deterioration of CYPA in E4 people. And in E4 people, when they do PET scans and the 20 to 40 age group, you're able to see deterioration of the cerebral microvascular system on PET scans. So they're not
20 to 40, you said, Dr. Green? 20 years to 40 years of age. It's already happening.
8 to 40 age group, they're able to see a deterioration of the cerebral microvascular system in E4 people on a PET scan. They're not showing anything else. They're not having any deficit in like memory or anything else. That's not happening until 60. But in the early age, they're seeing a deterioration of the cerebral microvascular system. So that's not like on PET scans.
Dr. Green.
So that can be my excuse, Dr. Green.
I have a question. So I'm wondering about the diabetic connection to Alzheimer's disease too. Is rapamycin working only in the mechanistically, only vascularly, or is there a backdoor?
Diabetic connection is an mTOR connection. Basically, mTOR is driving the disease. So mTOR is driving the disease, and anything that increases mTOR accelerates the disease.
Okay, so it's basically whatever rapamycin is getting to first is.
It's very important to understand what you bring up, a very important question about what's going on with diabetics. That can increase your risk of Alzheimer's disease threefold. But what it's doing is the diabetes is resulting in higher levels of glucose, but mostly higher levels of insulin. Insulin is the number one stimulation of mTOR. So they're spiking mTOR. So they're accelerating the disease process. Because mTOR is accelerating the disease process, that any sort of like disease that increases mTOR accelerates all the mTOR-related diseases, which definitely is including the Alzheimer's disease. So it's not this glucose is doing it, it's the glucose effect on mTOR, and the mTOR is driving the disease. Okay.
I guess I was wondering what came first, chicken or the egg, if that makes sense.
I think they're in parallel. They're happening in parallel when mTOR is higher, right? You're saying that it affects both.
Anything that's driving mTOR is driving the diseases. As far as like diabetes, they're having sort of like a problem with insulin resistance. The insulin resistance is now increasing mTOR. Insulin resistance is now increasing insulin, and the insulin is increasing mTOR. So you can think of all these sort of like things from like an mTOR-centric point of view. How is what's going on impacting on mTOR? If you're doing something that makes you very insulin sensitive, that means you have low insulin, you're not stimulating mTOR, you're very healthy. Anything that's sort of like anything, and you can think of just about anything through the mTOR pathway, is what's going on, increasing mTOR or decreasing mTOR. So that's sort of like you can look at all these things that way, but diabetes and insulin resistance, the number one thing that's going to be increasing mTOR and accelerating all these mTOR-related diseases.
So from that standpoint, it sounds like the chicken, well, which came first, the chicken or the, it sounds like it's the insulin resistance that's increasing mTOR and thereby impacting the blood-brain barrier or these other things that are impacted by Alzheimer's disease.
Well, so whenever you have anything that's increasing mTOR, you're impacting the blood-brain barrier and causing everything to be breaking down. Okay, let's have the next diagram.
Okay.
Okay, so this diagram comes from a study, a 2019 study called Rapamycin Alzheimer's Disease, Time for a Clinical Trial. And the paper was by Matt Kablin, who had been a top researcher in Alzheimer's in mTOR, in all things related to mTOR for the last 30 years or so. And Veronica Galvin, who has been a top researcher in Alzheimer's disease and mTOR for the last 10 years. In 2010, she was the first person to show that rapamycin could, in some studies, be preventing the development of Alzheimer's disease in some animals. But anyway, so she's the leading sort of like, the point is, Veronica Galvin is the leading person in the sort of all the studies related to rapamycin and Alzheimer's disease. And in this sort of like study, they had this one little diagram, and they were basically showing like different things. I won't go into them, that like they're saying mTOR could be affecting. And then they were saying mTOR could be affecting like high levels of mTOR, Alzheimer's disease, vascular dementia, normal cognitive aging, dementia with Lewy bodies, very interesting. That's the part related to like Lewy bodies is related to like Parkinson's disease, and what could be called a mixed dementia, vascular and Alzheimer's disease. So they're saying mTOR is having an effect on all these different things. And basically, in the study, they listed a dozen studies showing that first they discussed how rapamycin was real effective in like aging in general. Then they had a dozen studies to show, preclinical studies showing how rapamycin was looking very good preventing of Alzheimer's disease. And that included a number of the studies that I already mentioned. But the idea is that they were saying well, rapamycin is looking very, very good. It looks like the most number one thing that would prevent Alzheimer's disease. But nobody's done a clinical trial, and somebody should do a clinical trial. And they said, well, nobody is sort of like Big Pharma's not going to do a clinical trial because it's a generic drug. But somehow or other, they said like that NIH should come up with money for a clinical trial, or they said the money should come from some private people. Well, I thought doing a clinical trial was a real good idea, and that's exactly what I did. A clinical trial to see if rapamycin was stopping the development of Alzheimer's disease and mTOR and people who are E4 positive. So we don't need that diagram anymore. I'll just go over my study. My study could probably sort of like be $100 million if somebody wanted to do it right and get sort of like PET scans and CAT scans and psychological testing and everything else on everybody before you took the test, and then after like intervals, and then at like a year, and then 2 years, and then all sorts of like neuropsychological testing, and then all sorts of like special researchers to be going over all the data and like analyzing all the data. That would be one kind of a clinical trial. I had a much more simple clinical trial. My simple trial was simply, I saw patients. I saw patients since March, since April 2017. Everybody I saw, I measured whether they're E4 positive or not E4 positive. Right now, I've had close to 1,400 patients. There was about 700 patients who were double E4, and about 700 patients who were one E4 gene, and another, say, like 700 which did not have any E4 genes.
Wait a minute, Dr. Green. You just said you had 1,400 patients that you're treating, and you said 700 had double E4 and 700 had one E4.
700 have an E4 gene, 700 have no E4 genes. And within the 700 that have an E4 gene, there's 100 that have two.
Okay.
So it's like 700, all the E4 people together, of which there's 100 double E4, and there's another sort of like equal amount of about 700 who don't have any E4 genes.
I would think that anybody who had two copies of APOE4 would be looking for anything they could find to help them. And I would think the proportion of
98% of people who have double E4 genes are not looking for anything to help them because they don't know they have two E4 genes.
Right. But you would
And they
think
don't know
that
about rapamycin.
well, within our family, clearly Alzheimer's disease ran on my father's side of the family. It was very clear because people were affected in their 60s. And it just, when you see somebody deteriorating like that in their 60s, you go, there's something wrong, and I need to figure out what I can do to either to help them and or do something so that I won't, end up in the same fate.
Oh.
And it just surprises me that there aren't more people. I mean, I know there are people who don't know they have them, but
what you mentioned is very intelligent people who are very perceptive, look at what's going on in their family history, look at what's sort of like, look at a history, and if they have a history of dementia, especially, then they want to know, they investigate a little, they quickly find out that the risk factor is APOE4, and then they want to know if they're E4 positive.
right. That's what I did.
The problem is most people are not aware that there's any treatment. So people who are not aware of any treatment are basically inclined to say, well, I don't want to know. I don't want to have this black cloud hanging over me. There's nothing you can do about it. I don't know what to know about it. That's basically what the Alzheimer's Association tells people. The Alzheimer's Association tells people, don't bother being tested for E4. Why would you want to know that? That's what neurologists tell people. Why would you want to know that? You can't do anything about it. The number one purpose of this webcast is to let people know that there is a secret, that there is something that you can do about it. So that's
Such a horrible disservice to the world.
So my sort of like observations of my patients, following them for up to sort of like 6 years, having sort of like a wide range of people from 40 to into late 70s. My impression was E4 people do not have any more cognitive decline than non-E4 people when they're on weekly rapamycin, such that I could detect by talking to them. In other words, and it didn't matter whether they had a single E4 or whether they were a double E4. My impression was, in talking to everybody and asking how they're doing, is that people were not having cognitive decline. People were not complaining because I was telling people rapamycin is effective. So they were sort of like saying, using rapamycin should be effective in preventing cognitive decline. And I'm sort of like having problems with my memory. Or I'm sort of like not doing well. Or I'm sort of like very like not happy with how I'm doing. I'm basically not getting complaints like that. I'm not getting complaints from people saying, yeah.
Dr. Green, have you had any patients with symptom onset that have reversed or improved that you've noticed over time?
People very frequently improve. In other words, half the, like on a questionnaire, half the people said they were thinking better, but that wasn't specified whether E4 or not E4. But E4 people very frequently report that they're thinking better if they're having levels of confusion before they start rapamycin, and then when they start rapamycin, they're feeling like fine, and they're not having like any kind of like fogginess. They can tell what they're doing. My impression is that in the early stage of Alzheimer's disease, intelligent people have an extraordinarily good idea of what's going on with their brain. They can tell if they're thinking clearly, or if they're having some impairment, or if they're getting a little confused, or if they're not remembering things well, or if they're getting confused with different like things that they said I should be able to do this and now I'm not able to do that. If they're problems with their business, problems sort of like understanding conversations. They're very
That was my dad. He knew, and he talked about it all the time. And then his sister, my aunt, it's kind of, she just went into full-blown Alzheimer's disease without ever even mentioning it, you know? Whereas my dad talked about it for
people
15 years. I mean, he thought he got it in his 50s,
people can hide what's going on very well, and like sort of like they can carry on a conversation because it's not apparent if you ask them specific things like and like they say things like, I mean, they say it becomes apparent if you're with them when you tell them things they don't remember. You can see they're not remembering things you tell them, but mostly it's apparent to the people. The people have to sort of like tell people, I'm sort of like having, I'm worried. I don't think I'm thinking as well as I should be thinking. And they sort of like, most other people don't want to admit to them something like that. So they'll go along and sort of like covering it up until suddenly they're wandering around the neighborhood and it's clear they have dementia. But until they're sort of like,
that's crazy.
Around the neighborhood, it's not so clear that they're having dementia.
Yeah, I think psychologically you don't even want to admit it to yourself, probably.
Yeah, it's just, yeah, you don't want to admit it to yourself, but people have sort of like, they have anxiety about that. They have sort of like, they could tell, and it's a case of, what are you going to do about it? Are you going to tell people about it? Oh, well, he's going to, tell people about it. It's just a case of whether you think you can get any help. If you think you can get help, you'll be looking for help. If you don't think you can get any help, you're not going to want to be sort of like talking about it. And your family are not going to be talking about it. Everybody's going to be acting like so and so, like, and grandma is just fine. And like the kids are saying, what's the matter with grandma? They're saying, grandma is fine. It's like,
Right. And some people don't even, I'm recently with some friends of mine whose father is clearly, I mean, to me, it looks like classical Alzheimer's disease. And they just really, they're like avoiding the whole talk. They don't want to talk about it. It's almost like they think if we say it, then that's what he's going to have. Whereas if we ignore it, then he doesn't have that. And it doesn't,
that's the
I don't think that way. I was looking for whatever I could find. As soon as my dad, and as soon as I knew that was happening to my dad, I was trying to find things that might be able to help him. And so I looked into that for many years before I actually started, thinking about what was going on with myself. And I got tested, and luckily I only have one copy. I guess either my mom could have maybe had a copy too. We'll never know. But I just got one copy, and I'm pretty sure that was from my dad, who I am almost positive had two copies of the. And Michelle's mom has two copies of that gene as well.
I would say the thing about my study of like my observation of these 700 patients is it's not a standalone study. It's not like here's rapamycin is this. It's a study that's sort of like which should be viewed with this enormous amount of preclinical data, this enormous amount of sort of like preclinical data that indicates, yes, it seems like the preclinical data seems very, very, very suggestive that rapamycin should be protective. Then when you look at people that are taking it, then it could confirm, yes, I'm not seeing any cognitive decline in these people that I could detect compared to other people. They're not, I'm not noticing it. They're not noticing it. Although I would mention that I had two cases in which there were people who I saw in 2018, and they either knew or didn't know that they had APOE4 positive. Then they stopped taking rapamycin for whatever reason. Generally, somebody told them it wasn't a good idea. Somebody discouraged them. So for whatever reason, they stopped. Then I heard from them like 4 years later, and they were suffering from dementia. But these were the only two people in my study who were suffering from dementia, and these were the people who stopped taking rapamycin. In other words, they came, I saw them, they stopped taking rapamycin. They had a period of 4 years, and like in their 70s, and then they present with cognitive decline. So like the people who were actively taking rapamycin the whole time, none of those people are presenting with cognitive decline.
Did the people that you're talking about see improvements once they integrated rapamycin into their?
Generally, I don't think I wound up seeing them after that. The point is complaining about the fact it was just very, very noteworthy. Like, wow, they're having, they found out that they're E4 positive, and like, wow, those are the only two people I know about, and they were the only two people who I knew about who weren't getting treated. So the next topic I wanted to talk about is who's at risk. In other words, the idea of prevention. So the idea of prevention, it starts with knowing your APOE4 type. So from the point of view of prevention, I would say there's like three basics. There's like people who are double E4 positive. This is 2 to 3% of the population. These people, if 2 or 3% of the population, they may present with as much as 10% of serial cases of AD. They present to sort of like 63 to 75. They were very, this group needs absolutely intervention because they have about a 15 times greater lifetime risk. I mean, it's possible that they can live to old age without developing dementia, even if you're double E4 positive, but they have a markedly increased risk and a high risk of developing dementia at a young age where the sort of like consequences are absolutely like catastrophic. So these are the sort of like people who have to know the double E4 people.
Dr. Green, I read an article a long time ago, but it stuck with me. It said that the average age of onset for a homogeneous APOE4, so 2 copies, was 60. Average age of onset was 65 years old, and 60% got it at that age.
I think that's.
And then at 75, that was the average of age of onset for APOE4, and you had a 40% chance of getting it at 75 with a single copy of APOE4. And then it was for 85 was taking into the normal, it was more the average age of onset for somebody who didn't have an APOE4 allele. And they had a smaller chance. Like, I don't know, it wasn't, maybe it was 15% or 20%, but the 60% at 65 for double and 40% for the single carrier at the age of 75. It was in a little chart, and I remember it because it was like, wow, that's a big percentage of people that get Alzheimer's disease.
Yeah, I think that's the studies are like very, those numbers look a bit high. I think the sort of like median age of onset of double E4 people is 69.
Well, it made sense to me because I'm pretty sure my dad got it in his 50s, and my dad was dead by 72, and my aunt was dead before she was 70, and she never smoked. She wasn't particularly overweight. She did have
type 2 diabetes, though.
She did have type 2 diabetes, yeah. And she probably didn't manage it well because she was very laissez-faire about life and didn't worry about a thing. She's the one who moved into the Alzheimer's disease without ever saying that to us, even though her older brother died from it early, and he talked about it since he was 60 years old. So for 12 years, he knew he had it. It's just very interesting how different people are.
One thing that can change the statistics is when they use for the time of diagnosis. In other words, they might not diagnose somebody as having Alzheimer's disease until they have a fairly severe degree of dementia. Or they may say you have Alzheimer's disease when you have the earliest stage. So it might be somebody who's double E4 who at age 65 is having some sort of like decline, but really isn't really much like markedly impaired until age 70. So they might be called 65, it might be called 70, but basically the idea is that the people, like, in other words, it's like the chances of getting it are certainly from about maybe a 15 times greater risk if you're double E4, maybe a 3 times greater lifetime risk. But the much more important part is that the age of onset. The age of onset is the critical thing. And one sort of like figure that I thought was a very impressive figure is that they said the great majority of cases of dementia in people under 80 are people who are E4 positive. So just like 20 to 25% of the population, they're the majority of cases of dementia who have onset at 80 or less. And it's the people who are not E4 positive who are having an onset at a late age. And the impact is totally different. People having an impact in old age frequently survive 6 months and they're dying, as opposed to people who are winding up in a nursing home for 5 years.
Dr. Green, do you think that rapamycin affects these two groups differently? One better than another?
Which two groups?
APOE4 carriers and normative age-related dementia or Alzheimer's?
It has a very dramatic effect on E4 people because it prevents the breakdown of the cerebral microvascular system in the blood-brain barrier. What it does is sort of like non-E4 people, it's just very unclear. In other words, I don't have enough people in the sort of like 85, 90, 95.
That's interesting. Related to that too, do you think rapamycin can offset poor health choices in terms of, like, we know certain things contribute, like alcohol, like poor diet, heavily processed foods, lack of exercise, all these factors that contribute to your risk of getting Alzheimer's. Do you think that rapamycin can offset those, or you just need to throw the kitchen sink? If you have APOE4, you need to do everything possible, not just some hack like rapamycin.
Rapamycin is real helpful, but it still is important not to do anything that increase your risk. So it's like, in other words, like alcohol or being sort of like diabetes, things that increase your risk is definitely not helpful. I try to have all my people, especially adult E4 people, being insulin sensitive and having a very, very good lifestyle about sort of like what's going on. I think all these things increase your risk. Rapamycin is still preventing it, but it's sort of like it's not something you want to be increasing your risk factors. But to sort of like, you asked a very good question, what it's doing in people in old age? And basically, I don't know. It seems like that rapamycin would decrease their risk. But as far as having data, I don't have enough people, 90, 95, and 100, following long enough. In other words, I'm not sure, like, I don't have people in my study that are 100 years old and are cognitively intact. And I could say, well, you look at all these people. In other words, you'd expect people who are 100 years old, very, very few to be cognitively intact. So if I had a number of people who are 100 years old and cognitively intact, it would be very, very strong evidence that rapamycin was preventing dementia in old age. But at this point, I don't have enough data for that. I just have like very, very strong data regarding E4 people.
And that's another problem with doing a clinical study on people for this stuff, is they have to, they're expensive and will have to last a long time.
Yeah. But the bottom line I would say is that people should know that there's a way that you can prevent Alzheimer's disease if you're at high risk because you're E4, and that everybody should know the APOE type.
Now, Dr. Green, what about somebody who isn't particularly health conscious? I mean, I definitely have problems with wanting to eat like carbohydrate, sugar, not like soda pops, but desserts and things like that. And I definitely don't get enough exercise, and there's all these kind of things, but it's still going to be helpful no matter what, even if you aren't doing everything right.
I think two things. I think rapamycin is dramatically helpful regardless of what's going on, because it's affecting the cerebral microvascular system. But on the other hand, things that increase your sort of like mTOR make it harder to treat it, especially like the extent of having prediabetes and stuff like that, and diabetes. and sort of like all those other things to like make it harder to cheat. It's so much sort of like better to sort of like take rapamycin and decrease all your risk factors.
Mm-hmm.
Oh, so I wanted to mention the other thing. The people who should be sort of like alert to like testing, the people who have, of course, a family history of dementia, but anyone who has a premature history of cardiac death. In other words, maybe you saw like somebody died at like, 50 years old, or 60, or 55. That's suspicious for E4 because like
Mm-hmm.
people presents a premature cardiac death. And then anybody who has elevated LDL, that's a little suspicious. People who I see whose LDL without any treatment is under 100 are very unlikely to be E4 positive. But if your LDL, E4 people are generally elevated LDL, it might be 110, it might be 180, but it's usually not 80. It's not 90, it's not under 100. So those three things, elevated LDL, premature cardiac deaths of a history of, or anybody with dementia in the family, those are people who should be tested.
So there's a lot of discussion around LDL and how it really isn't a very good marker, and people really should have ApoB tested.
Oh, coronary artery disease. Yeah.
Yeah, for that ApoB is a better measurement of cardiovascular risk over standard LDL that everybody measures.
Prevention of coronary artery disease, that's another topic for another day.
Okay,
I know, I
all
was just
right.
gonna say, we're getting into a whole nother topic.
Okay,
Which, speaking of, we're about an hour and 20 in. Maybe we should
wrap up.
bring this thing.
Okay.
Okay, so to summarize, Dr. Green, how do we let people? Well, I guess we need to try and get awareness out there for people who have APOE4 that there is a treatment, and they should get tested so they know whether to take the treatment or not.
We should make a podcast to tell everybody about it.
Okay, we will. Well, we will. Well, we are, but okay.
If only.
Well, we're trying.
All right.
All right. Well, but bye, Raptors.
Bye, Raptors.